SeqMan NGen's 16S workflow is used to identify the presence of various species in a complex sample based on short regions of ribosomal RNA. This workflow will show how 3 different DNASTAR applications can be used in a continuous workflow. First, we'll use SeqMan NGen to assemble the 16S samples against a template. Next, we'll open the results in SeqMan Pro and export the 10 most populous species to Megaline. In Megaline, we'll align the 10 species and create a phylogenetic tree. After we open SeqMan NGen, we begin in the Welcome screen. Here we'll click Create New Assembly Project and then Next. In Choose Project Type, we want to do the metagenomics/16S rRNA assembly. And in Choose Assembly Type, we'll leave it at the default of templated assembly. In the Setup Project Files screen, we need to enter a project name and choose a project folder in which to save our results. The Input Biome Genome screen is where we need to add our 16S rRNA reference sequences. Reference sequences can be downloaded from any 16S rRNA database. The best known of these are Silva, Green Genes, and the Ribosomal Database Project. Here we'll use an enterobacteria database downloaded from the RDP website. To add this file, I'll just drag and drop it. In the Input Sequence File screen, we need to choose our read technology, which is Illumina greater than 50 nucleotides. And I'm using Illumina paired end data, so I'll drag and drop it to the Setup Paired Reads area. For the insert size, I'll leave that at the default and click OK, and then we can go to the next screen. I'm going to leave the assembly options and the advanced assembly options at the defaults, and now the assembly is ready to begin, so I can click the Assemble button. After our 16S project assembly is complete, we can drag the .assembly folder onto SeqMan Pro to view the results there. And next I'm going to expand the 16S project assembly window and use this pane divider, drag it down all the way to the bottom so we can view the top pane more clearly. To sort in order by the most populous sequences, we'll click on the Seqs column twice. And here we can see the 16S sequence matches from most to least populous. I'm going to select the first 10 of these and then export to Megaline by choosing File, Send Consensus to Megaline. So in Megaline, I'm going to do a multiple alignment by going to Align and choosing Biclustal W method. Finally, we can view the phylogenetic tree by going to the View menu and choosing Phylogenetic Tree. This shows how our 10 most populous enterobacteria species in our sample are related. To identify the species of enterobacteria, all I need to do is jot down the numbers, for instance the number ending in 356923, then go to my bacterial genomes that I downloaded from RDP and open them in a text editor and do a search for that number. And in this case it's Corynebacterium halotolerans.